Alrighty. So again, my name is Jikin Lau, and I'm a postdoc for Oscar and Dave at Texas A&M University. And today I want to kind of talk about comparing QTL versus genomic selection in a breeding context. Now, I do have to give a little bit of a disclaimer. I'm relatively new to genomic selection, so my background's more in QTL mapping and stuff. So I've just started looking at it like this past fall. So there's like any glaring things that are wrong, please let me know. Jeff, if there's anything glaringly wrong, let me know. So why study this topic? So I kind of have to tell a little bit of a story to kind of why I ended up comparing these two. So a little bit of background. We made two interconnected mapping populations that have been talked about last year. We talked about it in computational sessions, but the crosses were made in 2016. And they were Stormy Weather by Bright Eyes, which had around 200 individuals, and Bright Eyes by My Girl, which had 160 individuals. And we studied them for a lot of different traits. We looked at things like black spots, Cercospora, defoliation. We looked at other things like architectural traits, length, width, height. We looked at things like flower intensity. But for today, let's just talk about black spot, Cercospora, and rose rosette. So black spot is a fungal disease, and the picture of black spot is to the very far left. You have feathery black lesions and bad infestations which defoliate the plant. And you get chlorotic leaves. It just looks really bad. In Cercospora leaf spot is the second picture. Is circular lesions. They kind of look like a target with a necrotic center. And rose rosette, probably one of the worst diseases in the US for roses. Actually, these populations were made to study resistance to rose rosette. And those two pictures that you see on the far right are examples of what you would see in a rose rosette plant. It is, so rose rosette disease is what we see phenotypically from something that has been infected by rose rosette and Mara virus, which is transmitted by a little teeny tiny mite that's called Phyllocoptes fructiferus. And it is such a tiny mite that it is about a third, the width of the mite is a third of your hair. So it's teeny tiny, it gets blown around in the wind, and it hides on the plant, and it just, you can't really see it, but it affects plants. So these two populations that we built, let me go back up. So we built these two populations in 2016. We phenotyped them for a bunch of different traits. We genotyped them, and we did a bunch of QTL studies on all these different traits. And so last year, about last fall, Oscar and Dave came to me and said, hey, you've done all these QTL studies. Give me a list of individuals that are carrying the QTLs so that we can use them for the next round of breeding. So this being woody ornamental, it's like a breeding cycle, like 5 years. So I had to come up with a list of individuals to propagate so that would be used for breeding later. So there's two main paths that you could take. If you had populations that had genotype and phenotype information, you have multi-environment trials, multi-year trials, you could go the QTL mapping method or a QTL mapping way, which I had already done, or you could go the genomic selection route. Now there's pros and cons of both. QTL mapping, you need a linkage map. So that takes time and effort. You need to do the QTL mapping, that also takes time and effort. So it takes a little bit more, I guess, hands-on work. The genomic selection method, you basically just provided the genotypes and you give it the phenotypes. And with the G matrix, you're able to basically get a better estimate of your each phenotypic mean or phenotypic block. So from these two methods, you could select individuals for the next round of breeding. Now, there's actually a third way, which most breeders use, which is just phenotypic selection. So you use your mixed models, you don't really take into account genotype, and you select your best individuals. And using the QTL methods, so how we've done it in the past and how I did it previously, I did my maps in PolyMapR. Then I imported the maps into MapPoly to get genotypic probabilities and haplotype probabilities. And what you're seeing in the picture on the right is a picture of the haplotype probabilities of one individual for QTL. And in the QTL methods, what we did was I used a software that Chris developed, ViewPoly, to put in my QTL scans and put in my linkage maps, and I could go and select for the QTLs and the haplotypes that I wanted. So in the bright-eyed migro population, we isolated 10 individuals carrying black spot QTLs on linkage groups 3 and 5, Cercospora QTL on linkage group 1, and Rose Rosette on linkage group 5. In the stormy weather by bright eyes population, we isolated 14 individuals that carry black spot 5, Cercospora 1, and Rose Rosette on 5 and 6. Now, it's interesting that all of these QTL that we selected segregated in simplex to nulliplex. So it's expected that it's half to the fourth times 360 individuals. We're expected to have 22 individuals, and we got 24. So that fits the chi-squared expectation. So to kind of give you a better idea of, we saw this yesterday, Marcelo was showing us a figure much like this. If this black line going down the middle is where the QTL is, this is specifically a plot of linkage group 5. And if for this specific example, it's Rose Rosette disease, and if a individual carries the H allele or H homolog at that black line where the QTL is, then it carries the resistance QTL. So in this example, in this example, this individual does carry the QTL. So that's how we selected our individuals. We just looked through that big matrix that he was talking about yesterday, looking at position, looking at what homolog it carries. And that's how we got our 24 individuals for QTL analysis. So for the genomic selection method, we use stage-wise. And stage-wise, I used it to estimate GBLUPs and then create a breeding index from it. So in stage 1, I basically gave it the multi-environment, actually not multi-environment, it's one environment, but we took data over 3 years over the course of about 15 months. So each year was treated as an environment, and we took into account the months, and then we took into account blocking effects as factors. And then in stage 2, we added the pedigree information and genotypic information, in addition to the blues and the covariance matrix from stage 1. And then from there, we got our GBLUPs. And for each trait, I got GBLUPs. And what I did was I standardized the GBLUPs to allow for equal weighting for creating a breeding index. So kind of the scheme of how I made the breeding index had GBLUPs that for each trait that were generated by stage-wise. Then they were normalized to a mean of 0 and a standard deviation of 1. And I summed across to create a breeding index. And because this is, all 3 of these are disease traits, they're in the same, I didn't have to account for any different, like if you sum across the 3, the lowest is the best. So I didn't have to account for any signs. So this is just an example of before and after we normalized it. So on the bottom is the breeding index. I just needed something on the x-axis, so I put the breeding index there. And the y-axis is the GBLUPs that for each of the traits. The red being black spot, blue being Cercospora, and black not defoliation. That's actually supposed to be rose rosette. I don't know why it says defoliation. But after I normalized it, you see how the clouds kind of mash up together. It's one cloud now. So if I would have made my breeding index before normalizing it, that would have been really bad because you could accidentally weight something heavier, weight one of the diseases heavier. So that getting to a little bit of the results. So looking at just black spot, let me explain this figure first. So on the bottom, on the x-axis, we have the ranked genotypes, 1 being the best, 395 being the worst, with respect to all 3 diseases. So these are the ranked, basically, yeah, they're ranked. And something to note here, there's 395 here, where beforehand we were talking about only 360. So I had individuals that were genotype but not phenotype. So this accounts for it. So on this left pane, what we see are the ranked genotypes, this side being the best, this side being the worst, with respect to all 3 diseases. And on the y-axis is the GBLUPs of only black spot. Now, what's circled are individuals that only carry black spot QTL. So what we're looking for are individuals in this bottom left quadrant. So these should be the best, right? That's what we're looking for. And what we notice is that there are individuals that are missed. There's individual there, some individuals here that are not picked up just by QTL. Now, that middle pane, what we're looking at is the 24 QTL selected individuals. I kind of wanted to see where they plotted out to be. And we're the same. We're looking for this bottom left quadrant. And we see a lot more that are missed. So if I would have only done that, if I would have only selected the ones that carried the QTLs of interest, I might be missing a bunch of these guys. And looking at this one on the far right is basically phenotypic selection. I just used a mixed model to create a breeding index and selected the best 24. The only reason I used 24 was because I had 24 from the QTL side, and just to see where they fell out. And they fall out in that quadrant, but again, there's individuals that are missed, which is telling us, hey, the genotype information is helping. So for Cercospora, we see the same thing. And for rose rosette, we see the same thing. Okay. Now, this is kind of comparing apples to oranges, though, because on the y-axis you have one trait, but on the x-axis you have a rank that incorporates 3. So to be able to actually see these 3, you have to have it almost in 3 dimensions. So I hope this will play. So if you were to accurately show these 3 and highlight the 24 individuals, you want to look for the axes that is negative for all 3. And that would be this corner. Okay. So I'm going to play it and see if it will. So you see that group that's down here, this is where you want them to be. But there's a lot that are green that are not highlighted, red. So the red are the 24 that carry the QTL. And there's a lot of ones that are green in that corner that weren't selected. And this is the same for the phenotypic selected top 24. You're looking for this corner. And there are a little bit more here, but there's still ones that are green in that corner that aren't selected. So what does that tell us? That tells us that we can't just go with QTL mapping. We can't just go with phenotypic because genomic selection allows us to look at genomic selection isn't biased towards so QTL mapping is biased towards large effect QTL, right? And genomic selection kind of it weights things to the markers that have large effects. So you also pick up small effect QTL. So this is just another way to look at the data. On the x-axis is the rank from 1 to 395, and on the y-axis is the breeding value. And all I'm trying to show here is that the vertical lines, the first one is 20%, and the second one is 33% of the individuals. And at 33%, this one is, I don't know why I can't see that up there, but at 33%, you capture around 20 out of the 24 QTL selected lines, and at 20%, you have 15 out of the 24. However, you'll see when I zoom in on the next slide, I'll zoom in. There's a lot of individuals that are missed. On the figure on the right, that was for the phenotypic selection method, and at 33%, we have 22 out of the 24, and at 20%, we had 19 out of the 24. And as we zoom in, what you see is that there's a lot of individuals that aren't selected. So these have really high breeding values, but they weren't selected. Over here, this guy, and these all have very high breeding values with respect to all three diseases, but they weren't selected. So that tells us that, to go back to the story, if I would have only selected individuals based off the QTLs, I'm missing a lot. I'm missing individuals that have high breeding values. So to kind of end the story, we already selected individuals, we propagated individuals that were circled. So I need to go back and go get pollen from these kind of individuals and use them to cross, because they hold alleles that are important, but we just didn't know about those. So these are just some correlations of, on the x-axis is the GBLUP, and on the y-axis is the blues that went into QTL mapping, and just there's pretty high correlation between all the traits except rose rosette, and there's reason for that. It's because of scale and how much data we had. But other than that, in summary, QTL mapping methods help you identify which individuals carry parental haplotypes of interest. However, it's very biased towards large effect QTL. And genomic selection method allows for selection based off smaller effect factors, because it gives you an effect for every single marker. So the takeaway is, in a breeding program, we may need to incorporate both QTL analysis and genomic selection in order to make the best crosses for the next generation. And with that, I'll take any questions, if there's any questions.